welcome you back. And for today's lecture, we would be talking about the miniature blood vessels that we have, and these are the blood capillaries. Why I am discussing it separately? Because this is frequently asked in your exams. So, if I were to be talking about that, where these blood capillaries would be lying to any organ in the blood is coming, so it would be carried with the help of the arteries. Then we would make the arterioles, and from the arterioles, the blood would be going into their respective blood capillaries. From there, the blood would escape and would go into the venules, and from the venules, the blood would go into the veins. The idea is that only at the level of the blood capillaries we have got the cells or the tissues of our body lying, and it is only at this level where the exchange of the material would take place. Whenever the blood is there in the arteries or in the arterioles and in the venules and the veins, so the exchange of the material could not take place because the walls of these structures, they are very thick, so the particles, they cannot actually move out of the lumen of these blood vessels. This is only happening at the level of the blood capillary. So this is why I have written it down for you, said that these blood capillaries are important because at their level the exchange of the material would take place. And wherever the blood capillaries would be lying, so they would be making a kind of a network of the blood capillaries, and this is what we refer to this as the capillary bed. Now, if I were to tell you that how come the blood capillary structure would look like, so you just need to know is that unlike the arteries and the veins, they do not have three layers in their wall, but rather than they are only made up of a single layer of the greatly flattened cells. And these cells which make up the blood capillaries, so they were the endothelial cells. Previously, the endothelial cells were making the inner layer of the arteries and the veins as well, but the arteries and the veins, they would have the middle and the outer layer. But for the blood capillaries, this is only their endothelial cells layer which they have got in them. And the other situation is that for the cells of the blood capillaries, which are the endothelial cells, we will also have some pores in them. And these pores, they are referred to as endothelial pores or the endothelial gaps from where some of the components of the blood plasma, they would seep out. And that is important in terms of fulfilling the need or the requirement of the tissues that we have with us. So if I were to show you the knee diagram of the blood capillary, so here this is the blood capillary, and we have like taken some section out of here and we have presented it here. So if you were to see that these blood capillaries, they have a very tiny lumen in them, and their wall is made up of greatly flattened cells, and this is a single layer of the cell that I am showing you at the moment. And every individual cell that we have is the endothelial cell, and this layer is the endothelial layer, which is the only layer which these poor blood capillaries they have with them. Now, the idea would be that how come the structure of the blood capillaries is related to the function. Now, since I have said that at the level of the blood capillaries, the exchange of the material would take place. So for these blood capillaries, I have to increase their surface area. I also have to minimize the flow of the blood, isn't it? I also need to seep some of the blood plasma out of the blood capillaries for the efficient exchange of the material between them and the surrounding tissues that we have with us. So in order of their structures, the first thing is that these blood capillaries, they have a greater surface area to the volume ratio, so it would actually be enhancing the exchange of the substances. Now, as I have said, that wherever these blood capillaries would be, so they basically make the network of them, and this is the capillary bed. So when they would make the network, so you see that the blood has got multiple of the diversions from where it would be required to flow. So in this way, the speed of the blood is slowed down. And when the speed of the blood is slowed down, then as a result of that, more time would now be available for the exchange of the material to be taking place. Because if the blood was coming at a very fast speed in the blood capillaries, so not only it would be bursting them, but at the same time, less time would be available for the exchange of materials to take place. So since they have reduced the blood flow, so the time for the exchange of the material would be enhanced. The other idea is that in between the endothelial cells of the blood capillaries, now we have got these gaps in them, and these gaps, they are the endothelial gaps. The idea is that within the blood plasma, we have got the glucose and the amino acid and the stuff like that, and it has to seep out of the blood capillaries so that it can go toward the tissues. So thanks partly to these endothelial cells that they have these endothelial gaps in them, And that would allow the molecules to seep out of these endothelial gaps and then it go into the external tissue fluid so that the exchange of the materials could be enhanced. The other idea is that these blood capillaries that we have with us, so they have very thin walls. And when they have the thin walls, so the distance for the diffusion, it would be reduced. The idea is that the diffusion process that happens, so this is inversely proportional to the diffusion, inversely proportional to the distance that we have. So if I would increase the distance, the diffusion would be reduced. If I decrease the distance, so the diffusion would be enhanced. So now if I were to show you like these blood capillaries, now they are only made up of single layer of the greatly flattened cells, and here we have their surrounding tissues. So it is this much distance which the molecules they have to cross. If these blood capillaries, if they had got multiple layers in them, then ultimately the distance would be maximized and the diffusion would become less for the tissues that we have got. Now, the other idea is that these blood capillaries, their empty space that we refer to this as the lumen, so this is small. So when this is small, so this causes the blood cells, like the red blood cells, to squeeze through them. And when the red blood cells would squeeze through them, so as a result of that, the exposure of the red blood cells with the surrounding tissues would be increased, and by that, the oxygen released to these tissues it would be maximized. So this is how the structure of the blood capillaries was related to their function. I hope you would be clear with it.